YC grant. Thank you, Annie. We're going to be recording this between YC grant and NOHA. Today, we are lucky to have Sienna Correa Garcia talk about her work with LIMU in tank-based systems. Let me, for some reason, okay. Oh, shoot, sorry. Oh, such an amateur. Okay, just some housekeeping stuff. We're going to be recording the session. Please introduce yourselves in the chat when you get a chance. If you have any questions during the chat, during this presentation, please use the hand raising feature and we'll make sure that you have a chance to ask your question. Our next session is tentatively scheduled for April 12th. We're looking to incorporate UsingModernTechToRestoreLokoIaVideo to talk about some issues related to Lokoia and aquaculture. And then later in May, we're going to ask probably Ron Weidenbach to come back and give us a legislative wrap-up of the bills that are currently in the legislature related to aquaculture. So just to introduce our speaker, Sienna is a master's student at UH Manoa. She's in the marine biology master's program. And she works at the Waikalua Lokoia at the Waikalua Aquatic Institute, among many other places. And this is where some of her research that she's going to be presenting today was done. Born and raised on Oahu in Olapoko, did her undergraduate degree in Monterey Bay at California State University with marine science major and environmental science minor. And she's going to talk about cultivating native limu with land-based tank culture systems today. Some questions just generally, just to keep it in the top of your mind as she speaks today. Generally, how can we move forward with limu production, especially pests? How can we think about ways to approach pest control in limu production, and maybe some potential ideas for future research? And with that, I will stop sharing and I will ask Sienna to please start your presentation. Thank you, Sienna.
Thank you, Kai. Aloha, everyone. I will share my screen. Alrighty. So as Kai has mentioned, I'm currently doing some research at Waikalua Lokoia within our Pua Bootcamp or our Waikalua Aquatic Institute. Today, I will be presenting on my work on integrating indigenous fishery practices with modern technology and restoration efforts of native fish and limu. As I've mentioned, we are currently working at Waikalua Lokoia, which is in the south side of Kaneohe Bay. If you're familiar with the area, we share our backside of our Lokoia with Bayview Golf Course. And within this fish pond, we have a very special system set up, which is these tanks that we call Pua Bootcamp. And within these tanks, we have a system where we're hoping to continue to develop, and that's kind of what we'll be talking about today. And essentially, our tanks are getting all of our water from the Lokoia, which is very unique and special. And it's going through filtered tank or through a couple of series of filtration before going into a fish tank, and then to the limu tank before returning the water back into the fish pond. The idea of this aquaculture facility is to provide nursery grounds for pua or baby fish fingerlings to kind of prime them before introducing them into the Lokoia directly. They've had some trouble being able to just introduce them. So the idea is that they'll be able to grow in the water that they will be put into ideally and throughout the different Lokoia within the island as well. But having them housed in our tanks will obviously create a lot of effluent or fish doo-doo water. And we are utilizing the limu as a way to take out some of those nutrients, so as a bionutrient filter. And that's kind of what limu does in a fully functioning Lokoia. and just utilizing that resource. And we hope to grow a lot of limu because of all that nutrients that is coming from the fish before returning it back into the fish pond. Oops. So my two main objectives that I've been working on is determining nursery conditions that cultivates production of different limu species and expanding that literature in rigorous protocol for future generations to utilize. And that's really what we'll be focusing on today. But as a side note, my second objective is also to provide community-based local aquaculture learning opportunities, working with UsingModernTechToRestoreLokoIaVideo, having mentorship opportunities, outreach, and education as well. During the summer, we work with some students that help us out. So it's very exciting to be able to work on both sides. I'm really passionate about the two. So the mission of the research that we are going to be talking about today is to develop nursery techniques that are both accessible and affordable for nonprofit community partners to utilize in cultivating native limu. The idea of the Pua Bootcamp that is partnered with Sea Grant is to really establish this nursery ground that will be able to pass off on to PAF, who are the people that take care of Waikalua. And I guess just recognizing that nonprofit organizations and local IA in general don't necessarily have many resources all the time. So being able to come up with something that they'll actually be able to use and something that's really attainable for them and for other local IA to also create similar structures and systems in place in restoration efforts is really important. So the two species that we'll be working with today, or what we've been working with, is Limu lepeohina, also known as Limu ula, which is a halaminia, and also Limu monoeo, which is our Gracilaria species. So our lepeohina came from Uncle Wally's tank, or Uncle Wally's facility out at Afrak when we first started this area. And it's really cool to see it be so resilient through all the changes, but we'll talk more about that later. And Limu Manawea is from Met's program with Waianae High School and their aquaculture facility they have out there. So the three factors that we'll be looking at, or what we have been looking at, is light intensity, nutrient availability, and the maintenance of the limu. For light intensity, we filter the sunlight using shade cloth suspended over these tanks. So in the lighter on our left, we had 60% shade cloth, and on the right, we had 90% shade cloth. And essentially the idea, because we do not have any fish at the moment, we were trying to figure out in the absence of nutrients what we can do. So we really were just bouncing between the two tanks. And we'll talk about that in a second. So for light intensity, this is our daily light interval on the y-axis and looking at it over a period of a month, about a month. So the total daily light intensity is that top line where we actually use shade cloth for all of our experiments. So we were looking at 60% and 90%. And during this period of time, if you guys were around, we were really experiencing a lot of rain and cloudy weather. So some days our limu weren't getting very much sunlight at all, which is very important and something to keep in mind when we look at the results that we see. And as I mentioned, in the absence of our pua or our fingerling, we had to figure out what types of nutrients would be suitable to keep the limu alive and happy and getting the nutrients it needs to continue to grow rather than just survive. So we used a water soluble fertilizer and dripped it overnight into the designated tank. So 1 week we'll drip it into lepeohina at half filled, and then we'll let it fill up, and whatever was left over, whatever nutrients lepeohina didn't uptake, it would then flow into Manowea's tank, and then it would keep going back and forth every week. And with those 2 weeks, whatever week or whatever limu we were fertilizing, we were also cleaning it the morning of that day that it gets fertilized. And this was kind of the most efficient technique that we've came up with. It was a lot of work to figure out. We've tried so many different styles. And so we basically used a bilge pump and just recycling that water. And this technique was actually built off of the technique that Uncle Wally uses at his facility of just swashing around that water with the limu in it, knocking off whatever epiphytes that were on it. And having all that pressure kind of helped us keep cleaning it. So yeah, it's a very efficient system for what we're working with. And it definitely is a lot of work. So with the maintenance, looking at our shade cloth of between 60 and 90%, you can see on our left side of our 60%, both of Lepeohina on our left and Manuea on our right. With that 60% shade cloth, there's a lot of epiphytes or those unwanted, possibly diatoms or just some kind of other limo that's growing on top of it. And that really, we've noticed, kind of strangles the species of interest, so our Lepeohina and Manuea, and doesn't allow for the full potential of growth. Whereas on the right with our 90% shade cloth, there is not nearly as much epiphytes. And these were two different limus that were just randomly picked out of the batch, not systematically at all, just grabbed. And you can really see the difference just from these photos alone. So looking at our results, between the 4 weeks, we started off with the same amount of limo every week, or the beginning of our experiment, and looked how much it grew from the initial to the end. For both of them, we can see that our 60% of Lepeohina and Manuea both grew a little bit more than our 90% shade cloth. However, that might just be due to the epiphytes that we weren't able to get off of the limu, or it could have been just a little bit more growth because, like I mentioned before, some of those days we didn't have very much sunlight at all, and that might have affected how much these limu are growing. But also to keep in mind, these are both red species within the Rhodophyta order, so they do not require that much sunlight at all. So we really don't know what the reason for this is. But talking to the people that were cleaning the limu, myself and our Pua boot camp crew, being able to work with the 90% is a lot nicer because of all those epiphytes that I showed before. It keeps it pretty clean and keeps it a fast cleaning process. However, this is where we start to see some problems. The first limu that we worked with, aside from Lepeohina, was limu hulu vaina, and we would bring it over to our facility. We would notice it going from this really dark brown color, which you typically see, to more of this bright green color. We weren't too sure what was happening, and after a certain point, that limu will start to kind of disintegrate and wasn't really feasible for what we would want to produce in order to plant on other reefs, or also just to consume within our community. And unfortunately, after we wrapped up our project with our Lepeohina, it was beautiful, looking like this. And a couple weeks after that, we started to see our Lepeohina turn to start looking something like this, which is really unfortunate. We're not too sure if it's a seasonality thing. Maybe we just provided too much nutrients for it. Since it is a native algae, they actually don't require as much nutrients as some of our invasives or other limu does, because it's a deep sea algae. Yeah. So that's kind of what we're working with today. And mahalo for joining us today. And I'm really excited to talk stories with everyone and kind of see what you all have ideas. If you've had experience with any of this, we're open to everything and anything. Yeah, mahalo.
Right on. Great talk. Thank you, Sienna. That was awesome. So, as we typically do in these brown bags, we're just gonna open it up for discussion here. Does anybody have any comments, questions for Sienna, or any of the research that she's produced so far? I had a question that's, so I know that you identified the main pest as probably Ectocarpus. Do you know of any work anywhere else, possibly even outside of Hawaii, to control that kind of weed, other than physically kind of taking it off?
Are you asking me?
Yeah, sorry, I'm asking you, Sienna. Putting you on the spot.
Not that I know of. It really seems like if there's that nutrients available, they'll uptake it. So kind of controlling that nutrients. Don't really know what that looks like. I'll turn the question to everyone if anyone has any experience. I guess aside from all of this, something to plug into is I had no specific interest in limu coming into graduate school. I kind of came home, wasn't sure what I was gonna do, and I said, okay, if my other mentor, Malia Rivera, would guide me, I'll do it. But I wanted to make sure I did research that served the community. So I have no prior knowledge. I am learning as I go in 100%. And it's really awesome to meet with a lot of different people, Uncle Wally included, who's been guiding me through the way. So yeah, nope, no idea.
Awesome. No, thank you, Sienna. So it looks like Fred Mencher has a comment, a question.
Yeah, one way I gather you drip nutrients in constantly to your tanks. One way that sometimes helps against epiphytes is to do batch fertilization, where every couple of weeks you soak the seaweed in a fertilizer solution, let it uptake the nutrients, and then use low nutrient water the rest of the time, because very often the for example, Gracilaria can take up enough nutrient for a couple of weeks in a relatively short time, where some of the epiphytes aren't as good at doing that. And so it can help minimize the epiphyte growth.
I think that's a great idea. Thank you. I guess my question would be like, what do we think would happen when we do have that constant flow of nutrients? Because we'll essentially be getting it from the effluent water. Do we think it's gonna, because it's not like we can take the limu out, put them in totally just effluent water, and then put them back in. The idea is to have that flow through system. I'm wondering if there's ways that maybe we concentrate it by making it very like just half tank filled, maybe. I wonder if that would kind of help.
So that's a really good point, Sienna. So like in this sort of integrated multi-trophic aquaculture setting where you have constant, maybe even low level production of ammonia or nitrate from fish after they process the fish food and the effluent going into the macroalgae tanks, how would you manage, other than depriving the epiphytes of nutrients, how else might you manage it? Or how else, even with that situation, how would you manage it?
If you could find a nutrient that wasn't supplied in sufficient quantity in the effluent water, and use that essentially as a limiting nutrient. I don't know if that's possible or not, but it would be one possibility.
I tried to use my hand raising function, but it's not working, so I'll just chime in. I just want to add to Fred's comments. The process that he mentioned is similar to the process that Colette Machado was using on Molokai. I don't know if you guys have the Molokai limu growers manual that's described in that manual. So once a week, bringing the limu in, washing it out, and doing overnight treatment of fertilized water. And 24-hour, I don't know what the time period, but I'm thinking the next morning, put it back out into the fish pond. I have a couple of quick questions. Sienna, were you monitoring temperature and salinity?
We weren't necessarily measuring temperature and salinity. So that would be interesting, especially salinity for sure, because of all the rain that we experienced during that week. That was something we probably were assuming really made a turn for our Lepeohina.
Well, the reason I asked is because that question almost always comes up when we talk about growing limu in tanks, and what about the effect of rain on the salinity of the water? And for us, it's different. Well, for us, it's a flow-through system. So, for me, my thought was that rain, even if you get heavy rain, it's going to have very little effect on salinity. But that's just my just my thinking. So I was hoping that you would your numbers to show that it does affect, but does not affect. Yeah, talking about the rain. Number two, temperature, because one of the things that we learn at our limu tanks at the fisheries is that limu likes actually, I shouldn't say, the limu, sorry, I have to make one more correction. The limu, I don't think that's monoware. I
Yeah.
think that's the monoware loloa. So the difference is
Mhm.
monoware is Gracilaria cornucofolia. I believe that you are using the Gracilaria parvispora. So that has the name of monoware loloa. So very similar. So if you're doing a write-up, yeah, maybe double check on that name.
Yeah, we're actually gonna try and run some DNA tests because we also had our suspicions of if it was monoware or not.
Mhm. Well, I'll tell you what, but the reason I say it's not, it's parvispora is because of the branching pattern.
Mm,
I think you and I talked about that, the difference
Yeah.
between cornucofolia and parvispora. Yeah, just for me, it's like looking at the branching pattern. I guess that's it for now.
Yeah.
Thanks.
And I think we do have salinity and temperature measurements that other people on the team are taking, so it'd be cool to compare the two. I just didn't write that up in my data set yet. But
Okay. Oh, I know. Sorry. Final comment. The I'm finding that Gracilaria species like cooler waters. So, you know, we did a trial for 1 year, measuring growth rate for 1 year, and expecting that we would have better growth rate of that parvispora in the summer months, longer days, more sunlight, you know, more photosynthesis, so more growth. But what we found was that higher growth occurred in the end of the year, November, December months. Yeah, so cooler temperatures is preferred by the parvispora.
That's really interesting. Yeah. We were just talking about it today. We're hoping to run the same series in the winter months to see if we can compare that. It would be cool if our data cross collaborates and sees the same results.
Aloha, if I may add to that, thanks Sienna for your talk. My name is Simona. I work at Ocean Era and we also do a lot of limu cultivation. And, you know, similar issues like how many nutrients and things like that. But I think it's important to think about like the type of nutrients, like if you're doing a drip with a fertilizer, like what kind of nitrogen is coming in versus if you're doing an effluent, you know, that's mostly ammonia. So, you know, you have to think about that. Like, is it getting ammonia or is it like nitrate, nitrite? What we also find with our Halaminia or Lepeohina is that like tumbling is really important. So I don't know what you're like, was it tumbling a lot? Because that seems to lower epiphyte. It seems like if it's kind of more stagnant, then there's more room for the epiphytes to kind of colonize the limu. So thinking about that. And then the photos at the end I thought were really interesting. So it seemed like the limu was growing, but then it kind of crashed at the end. And yeah, just curious about that. The one thing, like before maybe we head into that, like the hulu waina, that looked like it was just kind of bleached out and starving in my mind. That's what I've seen with our hulu waina when we put it in the lab and it loses pigment. But if you give it effluent and you give it shade, you keep the epiphytes down, and then it'll, you know, it'll have like a really nice dark deep pigment when it's not starving. Like usually when it starts to bleach, it means it's like stressed, temperature, nutrients, something like that. So there must be some kind of a stress response going on with the bleaching.
Right. Thank you. Yeah, that was actually during the summer months. So kind of tying back to what Uncle Wally was saying, maybe because it was so hot, they probably maybe weren't happy too. I had a question for your Lepeohina. When you guys do, we also keep it tumbling constantly. Do you guys still see like that nice big blade form, or is yours more bunchy and kind of pokey, kind of like what we've been experiencing with tumbling?
It definitely changes shape depending on how you're growing it. It can even depend on the shape of the tank. What we
Oh.
find is that when we grow it attached on a line, we'll see that there's actually more branching that happens. So we'll see kind of like a more frilly branched if we like try to splice it into a line. When it's tumbling, we kind of get like more elongated branches.
Oh,
Mhm.
cool. Yeah, I was starting to look into that, like with porphyria or the stuff they make nori out of, how they kind of put them on the lines and it creates those nice big blades. So nice to know that's what you've experienced. So maybe we won't try that one anymore.
Yeah, but we're happy to talk to you offline as well. And yeah, good luck with all your work.
Thank you. Appreciate it.
Yes. This is all such great discussion. I feel like we should have a separate Limu brown bag seminar just for discussing Limu. I know Fred has his hand up, but let me just real quick, Tori added something in the text. I just wanted to see if Sienna, if you had a response to this. Could you add a different species in between the fish and Limu that might not use all the nutrients?
That's a great question. So in the diagram that I had in that IMTA system, we are actually looking into utilizing sea cucumbers and oysters as one of those filters that take out some of that nutrients before it reaches the Limu tank. And yeah, that kind of has a really nice connection of maybe that will take out some of that nutrients that the Limu wouldn't be able to uptake and provide it for the epiphytes instead.
Fred, you want to go ahead? Got another comment?
Okay. Yeah, one thing, Wally mentioned that at his facility, I guess the Limu grows best during the winter. But our facility was in Kahuku and we found that the Limu didn't grow very well during the winter and it grew extremely well during the summer. So it depends where you are. I think in Kona, they also find that it grows well during the winter. So, light intensity is important. We found the most light we could give it in Kahuku was the best. But if you're on the leeward side of the island, then maybe it doesn't need as much light, or at least it has more light than it needs during other seasons.
What species were you growing?
Gracilaria.
Oh, okay. Interesting. What was your, like, were you guys growing them in tanks as well, or was it in that kind of bigger?
It was in tumble culture. Yeah, it was a land-based system.
Cool. Very interesting.
We weren't using effluent from fish culture. We were just, in fact, when we had our well drilled, we had it drilled to a depth that would give us full strength seawater with minimal nutrients, with very little runoff in order to help us control the epiphytes.
That's really cool to see the connection of like, what's that perfect amount of nutrients where it's enough to keep them growing, but not too much that these epiphytes go crazy.
Yeah. And you could also experiment with different types of nutrients, how much nitrogen, how much phosphate, how much iron, various different kinds of nutrients that might encourage the growth of one species versus another.
Mm-hmm. That's awesome.
It looks like we have a little queue of questions here. Let's start with Ami. Sorry if that's how you pronounce your name, Ami.
Hey, yeah, thanks, Kai. Just had a couple mechanical questions for you, Sienna. We work mostly with freshwater. I do work with brackish water, but in our system, it's aquaponic. So we're doing the fish or other marine species, aquatic species, along with plants. So have you guys tried a system that uses a standing pond? So you go in between your fish and your plant area, and you're going to use it for treatment with ultraviolet C lighting in order to maybe tame down some of those epiphytes if they're in the water stream, water column. Also, too, you could try a mechanical scrubber that rotates like or maybe even just like a surface rub type of filter where you'll pass the water over the surface and kind of like use synthetic fibers to kind of try and catch the epiphytes on the way from the fish tank to this where the limu are, you know, with like a gauze or some type of like I don't know, synthetic fibers. You could use natural fibers, but I would suspect that synthetic fibers might be easier to clean. But something like that to help keep down the epiphytic transfer when you do introduce animals to it. Also too, I don't know if you've established nitrosomonas and nitrobacter cultures in there to do your nitrification cycle with, but once you establish that, because you're connecting animals to the plant tank, it's possible that, you know, you might have resident ammonia conversion through the nitrification in the limu tank, which will cause you to have changing levels of fertilization inside of that tank, even if you close off the channel. But and last thing, maybe you want to try and check out a topic called biofloc. Biofloc is a culturation of bacterial colonies. We use it mostly in wastewater treatment, as well as to generate bacteria that certain fish species consume as their food stuff. But it could also be used as a trap for your epiphytes so that they'll attach to those colonies or those medias that are inundated with the bacterial colonies. But just an idea of maybe throwing it off a little, giving the epiphyte something else. But yeah, thank you.
Thank you so much. I really liked when you're talking about utilizing some kind of synthetic fiber optic kind of thing or fibers to catch some of the epiphytes. I think that's a very attainable mechanism to kind of knock off some of those epiphytes, especially in areas where they would, or in, I guess, facilities that nonprofit organizations might be keeping and stuff like that. So that's really awesome. Kai, do you want to talk about our the, what's it called? Why am I blanking?
The recirculating
Yes,
system.
the research tank. Yeah.
Yeah. So yeah, thank you. I mean, yeah, we are actually just starting to sort of do some side-by-side testing with recirculating systems that have active biofilters that have the nitrifying bacteria that you were talking about to convert direct ammonia into nitrate or nitrite, and see maybe that might make a difference with limu growth. Yeah.
It could possibly. I use those types of gauzes in my grow beds in order to catch free-floating algal cells, so that, you know, they collect in one area and then they have kind of like their surface. I forget the name of the chemical, but they kind of attract one another into longer strings of algae. So it makes it easier to collect it into one space instead of it spreading out into a bed. So maybe that kind of idea might work for you guys. By the way, Kai, I love TR. I got to see it the other week when we dropped off the fish tanks. Loved it.
Oh, right on. Thanks, Ami. Thanks for your comments. And looks like next we have Maria Haas.
Yeah,
are you
aloha.
there? Yeah, go for it.
Yeah, Sienna, that was a great presentation. Thanks for sharing all that with us. You mentioned using oysters to remove nutrients from the water. Could you share some details on what you're planning to do with that, please?
So I actually focus on the limu directly. I don't know, Kai, do you want to maybe touch upon that?
Yeah, sure. You know, oysters or any kind of bivalves, clams, or, you know, any potential animal like that in the integrated multitrophic system. We were experimenting with all kinds of bivalves for, you know, to really perfect this IMTA kind of situation with limu.
Hey, thanks for that. You know, bivalves are kind of notorious for not doing very well in integrated systems. But that's just my 2 cents worth. But yeah, nice presentation.
Thank you.
So does and yeah, we're coming up on 12:40-ish here. Does anybody have any other comments or any other suggestions for this work moving forward? It's a great discussion. I really, honestly, I think we should have a limu brown bags in between all the other brown bags. It's just my opinion, but any other comments or suggestions? Or it's a huge group. Almost 30 people here. Great.
Okay, I have another question. Are you planning to continue this project? And if so, like, try to work fish into this, into this process?
Great question. Yes, we have fish en route to come to us soon. So our next, I guess, experiment would be to really focus on the fish and see how we prep the fish tanks and how that influences the amount of growth we get for the limu specifically. Using this study, we are set on using that 90% shade cloth was something that really influenced what we do next. And aside from working at Pool Bootcamp, I'll also be really looking into a controlled settings of having mason jars and filling that with one piece of limu, and really starting to get down into the nitty-gritty of how much nutrients it needs, how much phosphate, and how much, blanking on the other one that we'll be looking at, but those two nutrient sources, I think that someone mentioned earlier, as well as putting into account salinity and the temperature. So all of these different things that we're looking at large scale, did it kind of backwards. Now I'm going to take it really small scale because we do see there's definitely an impact happening. So that's the two, I guess, major projects that are coming up on the horizon that really hope to inform this study and other studies as well.
Well, anybody have any other questions or comments moving forward? Oh, wait. Yeah. Just all congratulations to you, Sienna, in the chat.
Thank you everyone for joining, and thank you for all these great ideas and suggestions. I need to come around more often. I really like these.
Yeah, and please, everybody, also thank you for your attendance and for continuing to support our collaborative and all the speakers. Again, we're gonna probably, if there's no other comments or discussion, I think maybe we'll wrap it up now. But again, please look out for mid-April for the next one. And maybe we'll call it a brown bag then. Thanks everybody. Aloha.
Thank you everyone. Uncle Wally, are you still here? Is he gone?
No, I'm still here.
Okay. I was just typing in the chat that I'll be coming